Now, let's get into what kind of methods to study nervous system. There are 4 general methods of studying nervous system. 1, case studies, and 2, screens, and 3, descriptions, and 4, manipulations. So, case studies are identifying naturally occurring events to develop hypotheses. So which case study means just, we have no control. It's naturally occurring. Okay. Screen is we perform search for anatomical structure or neuron, proteins or genes that could play a role in subject of interest. So we search asystemically to see if there's anything which can be important. And the third one is description. We just watch, okay, to observe the nervous system, without manipulating, without touching any variable, okay? On the other hand, manipulation is another way of studying, which means we want to test hypothesis by determining the effect of an independent variable on a dependent variable. So we modulate or we manipulate, and then we see the outcome. So that's also a good way to understand the whole system. So first method is a case study. It's a very, very famous story by an event that happened to a subject that demonstrates an important role for an aspect of the nervous system. What it was is, in fact, it happened in 1848. There was an accident to a Phineas Gage, and he was working in a railroad track. And there was an explosion that a big rod is passing through his eye and his left frontal brain. And that damages some part of him. And look at this is that diagram. And what it says is that injuries reported effect changes his personality and behavior over remaining 12 years of his life. So that's maybe giving an idea that the damaged area may be related to his changes in his characters. Okay, so this is case study. What's good thing about this? This, if you're lucky, often reveal very important information. Because in this case, frontal lobe of the brain, maybe because it was damaged and it changes his character, he's not no longer Gage. He's just totally different a person. Very before and after, very angry, anxious, and not too easy to work with. Okay, but the limit of this approach is usually not, yes, you can't make this happen again, okay? So usually not repeatable. And because of this, cannot precisely recreate it in a laboratory setting. So second, as a screening. So screen is a method to determine in nervous system, brain, let's say, which nuclei, what kind of neurons, what kind of genes, down to proteins are involved in a particular biological process. So by screening each of these, we may be able to identify some candidates that can form the basis for future, yes, hypothesis-driven research. Okay, let me give an example. So fMRI scanning of a subject in response to specific stimuli, or task to identify which brain regions are interesting. So you may know heard about fMRI, so-called functional MRI, showing neural activities lighting up when the subjects are doing something, okay, to specific stimuli. So for example, we are interested in brain region, which is maybe related to recognizing human face. And maybe we believe that our brain, there's a specific reason, maybe more related to house. So we show the subject different kind of face, and we perform fMRI, showing that which regions are lighting up or down. And response to houses, again, similarly, we do this, and maybe these areas are related to, to these external stimuli, so-called, in this case, different kind of face recognition or recognizing house. Okay, the third is the observing nervous system without manipulating or intact. It's called description. So testing hypothesis by determining the effect of an independent variable on a dependent variable is, we want to see, observe, the act of observing properties of nervous system without manipulation. So this is called, we call this science as descriptive science. So neurons can be described in terms of what genes or proteins they express, what kind of shapes do they have, do they have a big cell body or small tiny cell body, do they have a big dendrites protruding or smaller dendrite, what's their length of axons, so this kind of morphology, and how many neurons make up a population of neurons, are there a big chunk of population or small, and also their electrophysiological properties such as action potential. And we can describe this by looking at or measuring. Okay, so that's a descriptive science. And observation, excuse me, observation and description, in fact, form the foundation for understanding the relationship between structure and function of the brain. A famous example is, yes, Ramon y Cajal. So, however, there are tendencies that if you just say, I want to observe, describe, people may not want to give you money for funding because, hey, you have no idea, and it's like a fishing. But the most important thing, this observation or description forms a basis which can help formulating a new hypothesis. So this is fundamentally important. We have to admit that. At the fourth is testing hypothesis in neuroscience by manipulation, an aspect of nervous system or environment, and examining the effect of perturbation on a separate aspect of the nervous system. In engineering point of view, we have, let's say, some kind of radio system, some kind of mechanical system. We want to understand their system characteristics by sending an input, which is changing the input and see the output changes. So similarly in neuroscience, this nervous system, we want to disturb or manipulate one variable, which we call as an independent variable, and we observe the output, such as action potential difference, such as maybe gene expression difference, and that as an output or dependent variable. So manipulation experiment we are basically testing the effect of an X on Y. So in this case, X, yeah, is an independent variable, Y is a dependent variable. So here, this manipulation, I want to introduce a concept of loss of function experiment and gain of experiment. This is very important because of some variable, is it sufficient enough to cause an effect, or this variable is necessary condition for causing this. So these necessary and sufficient conditions are very important to identify the causal effect. So an aspect of nervous system is, let's say, increased relative to normal. For example, expression of gene or protein or electrical activity in brain region or neurotransmitters in extracellular medium is increased by doing something. We call it as a gain of function. So, but on the other hand, so those aspects of the nervous system is partially or totally disrupted. Let's say increase versus disrupted or lower down, and we want to see the output change. And that we call as a Yes, loss of function. So, for example, let's say we believe this gene is very important, okay? And so we deliberately lower down, knock down, or lower the expression of a gene, and then we find some particular output, such as behavior is a change. Then we can consider that gene is at least necessary for revealing that particular function. So then how can you know this? This is by luck or something else. So we can devise a way of gene, uh, gene expression down, and then we see the particular aspect of behavior is gone, and then we re, reveal, increase the gene activity, and we saw the behavioral changes recovered. So that is a very good way showing that necessary condition and sufficient condition, which is showing that the causal effect of this gene to this particular behavior A. And this is we call rescue experiment. So the aspect of nervous system is lost and deliberately returned, and see the output is also returned. And that's a good follow-up for the initial, yes, loss of experiment. So you can see many very good studies are actually using this deliberate loss of function and rescue experiment, showing that the particular gene or protein may be very important for this particular outcome. So now I want to discuss about correlation and causation. So we're doing science, we have to be very careful about our statement, correlation and causation. Oh, okay, I have missed this one, an example. A mouse that lacks a gene is unresponsive to painful stimuli. So I have a mouse and I deliberately uh, manipulate the mouse gene A is lost. And this mouse now turn out to be having no pain. So that's, how can we conclude this? A wrong conclusion would be this gene is regulating this pain detection. That's probably not the best way. Okay, so the right way is when we remove this gene and we see the mouse do not recognize the pain perception, so that means this gene is at least necessary for proper performance of this pain detection. That is more precise or proper conclusion. And why? Because this gene may not directly regulate pain detection, but it could be indirect so that the whole effect could be correlative, not directly causal. Or there are many other ways to regulate the pain detection too. Okay, so that's why this gene, we can conclude, is at least necessary condition for proper pain detection. Okay. So, so very important thing is correlation does not imply causation, except when it does. Yes, due to very difficult study, medical studies are a lot of times correlative, and it's very difficult to show reveal causality in medical clinical experiments because they involve humans, which is very rare. While in preclinical or animal-involved research, often we can get into more of a chance to do a causal study. So let me give you an example here. So some people say, hey, I believe cell phone is causing cancer. And okay, you believe that? Show me the data. And these are the data over time. This is total cancer incidence, growing this, and then cell phone users this. And there's, the time-wise, cell phone user increase do not necessarily support the cancer incidence rise. So just to be safe, until we don't see more data, maybe cancer causes phone user increase, because this is very similar to what we know our history of data of smoking and lung cancer. So for example, this is smoking, and we saw a decade or several decades later, lung cancer increase, which is a very correlative, but it gives some clear clue that this will increase of smoking may accumulate in the body to cause a lung cancer increase. Okay, so it's very difficult to definitely say one thing causes another. To do that, the cause is taken out. Does the effect still occur to the degree which it would have if the cause was present? So cause was present or out, we have to see the effect. And could there be any other causes that could contribute to the effect? So in this case, smoking, let's say, causes lung cancer, but it's possible that this is by chance. Maybe over time, there are other things such as environmental effect, maybe causing the lung cancer increase. So we have to be very careful to say A causes B, which is a causation. So that's why people value very rigorous research of revealing and proving a causality. Okay, as a summary, we discuss scientific hypothesis as a worldview that is testable and falsifiable. And we discuss about studying nervous system by case studies, screening, description, and manipulation. And we also discuss gain of function and loss of function experiment, and which gives a sufficiency and necessity. And to show a causality, we can develop a very clever rescue experiment and the difference between correlation and causation. And thank you for your attention.